Okay, why don't we get started, everyone? Thank you for joining us today for the fifth webinar in our 3Rs webinar series this year. Today's webinar is entitled Making Human Organs and the Path to Replacement. It's being presented today by Ping Ye, the co-founder and chief executive officer of StemoniX, Inc. And Ping is being joined by a few members of his team. Blake Anson, the VP of Business Operations. Glenn Smits, the Senior Vice President of Discovery and Early Development at Biopharm, a cancer genetics company, as well as Barb Jones, also a Senior Vice President of External and Regulatory Affairs at Cancer Genetics. So a couple quick reminders for everyone. Today's webinar is being recorded and will be made available to everyone as soon as possible after the system processes the video. The webinar was approved for 1.25 AABSB race approved continuing education credits. I know that there are many of you that are logged in sharing links and things, so if you are sharing a link, please reach out to me at leach@njabr.org to make sure you get logged for your continuing education credits. If you're logged in with the link that was emailed to you, the system will log your participation today, and we'll have everything we need to make sure your credits are approved appropriately. We will be taking questions throughout the webinar today. Ping will be pausing at various points to address questions, so please submit questions as you have them during the presentation today, and we'll get to them. We also have a more detailed Q&A session at the end of today's presentation. And you may also reach out to Ping directly at pingye@stemonix.com with any additional questions that you may have, and Ping and his team will be more than happy to answer any questions for you. Briefly about our presenters today. Ping is a nanotechnologist and engineer by training for the technology and storage industry. He received his master's and bachelor's from UC San Diego and his MBA from the University of Minnesota Carlson School. Ping built and led multiple teams and organizations that brought transformational hardware and software technologies to market. He is also a cancer survivor who redirected his expertise to finding safer and more effective medicines for patients. As I mentioned earlier, joining Ping today is Blake Anson. Blake is a seasoned leader who developed iPSC-based solutions in drug discovery, academic and government sectors over the past 15 years. He's also authored 30 peer-reviewed manuscripts and book chapters. Glenn Smits is the Senior Vice President of Discovery and Early Developments at Biopharm, which is a cancer genetics company. He has significant scientific and regulatory experience and expertise on bringing therapeutic assets to the clinic in cellular therapies, small and large molecules, medical devices, and combination products. And finally, also part of the team and joining our panel today is Barb Jones, who's the Senior Vice President for External and Regulatory Affairs at Cancer Genetics. Barb is a recognized leader in identifying future transformation technologies to rethink drug development. She also is a former advisor to the United States Congress and international health and regulatory agencies on science and technology. So we're very pleased that Ping and his team are able to join us today. With that, Ping, I'll turn it over to you to begin today's presentation.
Great, thank you, Tom. And just for clarification, Blake, Glenn, and Barb are on the panelist for today because, as some of you may know, we have announced a merger with Cancer Genetics, and Vivofarm is a cancer genetics company, and together we really look forward to being part of one team together. And so that's a little bit of background on that. And how is my screen coming forward here?
Looks good.
Excellent. Well, we have quite a bit to cover, and it is a pleasure to be here with all of you today. And I'm going to start with a thank you for all that you do for patients, because I was one of those patients. And 8 years ago, I was diagnosed with Hodgkin's lymphoma, a form of blood cancer. And it was a very difficult journey, but it wouldn't be possible. I wouldn't be here, and it wouldn't be possible for me to be here with all of the work that you do for patients and finding medicines for people around the world. So thank you. And I don't have an academic PhD, but I do joke that unlike the panelists that will be with me at the end, I have a PhD because I'm a patient had disease, and it wouldn't be possible again without all of your work. So thank you. Now, my journey through this process of becoming a cancer survivor was not a straight line. I was resistant to the standard chemo regimen of Hodgkin's lymphoma, and I had to take something that was more potent. And it was also more toxic potentially to my heart. And so that began the quest and journey of how StemoniX was born and how the company came together. And so today, the topics that I'll cover will be really what we've learned the last 6 years around this question of how do we find safer and more effective medicines. And as Tom mentioned, I have a background in engineering, but it was quite serendipitous. That background became very helpful to what we are doing at StemoniX. And so I'll cover some topics such as the recent history, and we're really entering this convergence renaissance around iPSCs, or induced pluripotent stem cells. And then number two, the application of these technologies and how it's transforming drug discovery, and really bring this convergent human first approach. And I'll go through some examples as you can see here, and we're very excited to cover what we've done over the last few years and give you a general insight into how this industry is working with this 3 R's principle, which is the topic of today. And then lastly, now and into the future, some examples of how we're moving forward today and how these will move us differently into the future around what we're doing here in drug discovery. So let's get started. Here is the quintessential slide that many kick off their presentations. But one of the things that we learned early on many years ago, as I started investigating this space, was that not only are there high failure rates in clinical trials, but that the clinical safety failures were really dominated by brain and heart related safety failures, almost 60%. And that really caught our eye in terms of if we were going to use induced pluripotent stem cells, which we'll talk about today, and then turn them into representative microorgans, well, maybe we should start in these 2 areas. And then it was also a great surprise that late-stage clinical failures were often due to lack of effectiveness. And of course, the long, costly road to get therapeutics to market. And so the observation many years ago was that, as we were hearing from key opinion leaders, were that the models that were being used were underperforming in terms of the effectiveness and safety predictions. And this could be, by many accounts, the result of cross-species differences, leading to really a lack of understanding around the disease mechanisms that lead to potential new therapeutics. And then lastly, we'll look at how we can rethink the whole drug discovery flow. And here the point is that human biology is introduced late, which in the past might be logical because these capabilities didn't exist, but now they do. So how can we improve this translation process? Well, I'm an engineer, and what I'm going to show you today would not be possible without the amazing team here at the company. And many of them have been on the forefront of this new segment of the industry in stem cell biology. And they've been some of the most published in the world with regards to what they do. And so we're very honored to have them as part of our team. And here's a quick snapshot, even some recent ones in the pandemic time that we're in. And again, this would not be possible without the team and the community that has supported us to this point. Now, the story begins really at this amazing time that we're in now from the standpoint of this convergence renaissance. And this renaissance of the fact on the left that we're able to engineer biology. And that ability to engineer biology, whether it's through induced pluripotent stem cells or editing DNA and RNA now, is really one of the sparks for this renaissance. And with that combination is computing and software and hardware combined to give us the processing, the storage, and the data science, augmented intelligence, machine learning capabilities that all come with that. And together, that confluence brings new ways of doing things. And in the end, for all of us, new discoveries. And so let's begin with iPSCs, or induced pluripotent stem cells, a technology that won the Nobel Prize in 2012. And the technology really brings forth the ability, and I believe it's one of the top inventions in human history, and it really is an ability to convert samples of skin or blood and convert that into a form of stem cells, which are pluripotent, which people call induced pluripotent stem cells. And this is through increasing the expression of the Yamanaka factors after the Nobel Prize winning scientist Shinya Yamanaka. And that capability from the previous screen to this one is really the beginning of this era of stem cell biologies, because then you can convert these induced pluripotent stem cells to things like brain cells. And here, this is something that we do in-house at the company. And you can see the structure on the left and the staining for synapsin in green on the left. But the neural network that's shown in the video on the right, this calcium measurement of the neural network, and can see the activity there that's possible when you differentiate these iPSCs into these specific organ cells, such as these brain cells. Now, there's complexity here. You're noticing here that the green are the neurons and the red cells are the astrocytes. So what's unique about our system is that it's a co-culture of cortical neurons and astrocytes. And that gives us some very interesting capabilities and characteristics of the model that we will talk about today. But here's a beautiful image that was taken by one of our scientists. Now, that was looking at a two-dimensional system, and what we're becoming known for are three-dimensional systems that you see here. So floating spheres of these cortical neurons and astrocytes, and these spheres are about 0.5 millimeter in size. Now, there's one thing to make one, but as many of you know, this isn't just about making one because we don't have one drug to bring forward in this process. There are thousands of potential candidates that we need to sift through to find the right clinical candidate to move forward into the clinical trials. And so you really need to have the scale and the volume to screen for that many compounds and variations. And so that's what we have done. Now, human modeling for industry and research, I want to talk a little bit about complexity and consistency. And here's a chart, and there you can see my daughter's all grown up on the right. But and I think we're having a laugh from my world famous dad jokes. But as you can see on the left-hand side, we're from tissue complexity to all the way to human beings. There is this spectrum. Now, throughput is also almost inversely to that, as you can see in the spectrum of complexity and consistency and throughput. So where do these human organoids sit? And they kind of sit in the middle, but we're now entering a time where not only do you have organoids, but now you have what are called assembloids, which are, can you take different forms and organ systems and organoids from different organ types and actually assemble them together. That's now in process and at its beginnings. There are microfluidic chips, tissue on chip technology. There is technology, mechanobiology, is controlling the environment and the materials and the microenvironment to actually shape the biology. In addition to this, there's also ways to increase the complexity of these models by introducing different diverse types of cells. And we're going to get into these technologies, most of them today, and just to give you an overall picture of complexity. And often complexity is at the detriment to consistency. And what we've done in the company is really enable enough complexity to answer key questions that you're trying to answer while having the consistency to get meaningful information from that data. And so recently we had a conversation on a panel with key opinion leaders from the NIH, FDA, Big Pharma, and it was consensus among the panel that these human models that are made from induced pluripotent stem cells, for example, are here to stay. So it's not a matter of if we're going to use them, but when. And today I'm going to share with you that when is now. And it's in a very exciting space, and I hope after this talk you're going to be just as excited and interested as well. So one of the things that StemoniX did over the last few years to create this new way of doing drug discovery is converging this human biology with a multidisciplinary approach. So it wasn't just biologists, stem cell biologists, it was really the convergence of various disciplines, from engineering and computer science and manufacturing. It all came together. So in a sense, the 6 areas that we really brought to this industry was this human high functional capability that also is around tissue structure and shape, the ability to shape that through things like mechanobiology, bring forth the engineering to create the consistency with the biology to bring about a level of consistency that you could do screening. And then the level of scale, because this is really key. The scale with iPSCs are possible because they are immortal cells. You can keep growing them, they keep dividing, and you almost have an unlimited supply if you do it correctly. So that is one of the key powers of iPSCs, is in a sense an unlimited inventory if you want to go that route. Now we've also added ease. So from a distribution standpoint, we can ship these all over the world at room temperature. So not with 10 pounds of dry ice, but we can get them all over the world at very low weight and low cost. And it became evident, and you'll see in the next subsequent slides, how important it is to have software at our fingertips and how we can integrate that with biology, because we're dealing with so much data. So integrating that is the next story that we will be talking about in this presentation. And then that integration leads to this validated prediction of compounds that the pharma industry is interested in. So this is a critical slide, and it's not because I spent my Saturday afternoon this weekend putting it together, but it's because it really shows how we can compress the timelines and achieve the 3R principles. So you have preclinical, clinical process, left to right time. Now you can see this build. What we're doing here with iPSC technologies and having these human models is the ability to have a human first approach way early in the preclinical process and compress that to have this human first insight to increase the probability of success, because you're getting human information from these models. And then it's also iterative to eventually get to the clinical candidate for clinical trials. And you'll see the build here, the iterative nature of testing therapeutics or recommended therapeutics iterating it through the process to get to higher probability of success therapeutics. Now, this is the addition of human models plus software, AI, and machine learning. And we'll get into all of this, but this really lays the groundwork. I really want to heighten that this lays the groundwork for our conversation today. And then really and this is about optimizing time, cost, and accuracy as we go through this process, but in a very compressed way by integrating biology and software technologies together. And then you can bring in the traditional seasoned approaches of in vivo testing at the end of that, so that from the standpoint of replace, reduce, refine, you're able to shrink it down. So you can see the build here. You can use fewer animals and in multiple dimensions, in time and in number. And that's critical to our conversation today. So I'm going to pause there and see if there are any key questions. Tom, shout out if there's anything that there's any dying questions out there. But now we're going to get into the application of these technologies, and we're going to give you many examples of how this is transforming the industry already. So I'll take a quick pause.
I'll give it a second. If anyone has any questions, please feel free to use your question box and send them over to us. We'll give it a second here to see if anything comes in.
Sure, no problem. Thank you. All right, I think there's a lot of slides, so we can leave it for the panel if that's all right, Tom?
Yeah, one just came in actually.
Oh, okay.
I'll read it to you. Can you give some specific examples of what type of animal studies would be replaced?
Yeah, one quick example would be like neurotoxin. That's actually one of the next several slides we'll cover. So maybe we'll jump right in. How about that?
Sounds good. Go right ahead.
All right. So laying the groundwork for how we can replace certain aspects of animal testing examples is laying the foundation here of how we engineer biology. So what we're doing, once again, at a very high level, is bringing together biology plus computing to rethink how drug discovery can be done and to find the safest, most effective medicines. So here you can see one of our plates. I'll show it up if you can see me on the screen here. One of our plates, you can put a wide variety of different platforms. Brain, heart, and pancreas are our 3 platforms. And we're generating so much data now that it is essential that we have the software technologies at our fingertips to analyze data, to rank order the therapeutics that the potential therapeutics, the compounds in our experiments, and then being able to cluster mechanisms of action, for example, to, in the end, bring forth the safest, most effective medicines using these technologies. So once again, the iPSC technology and the engineering of biology. So here are the 3 platforms, differentiating them into the different organ cell types, but that was really just the beginning. We took it many steps forward to now we can put at high consistency and scale into international standard microteeter plates, brain, heart, and pancreas. So let's dig in. And today we're going to focus a little bit more on neuro. And here you can see our neuro plate. And what you're going to see here is a video. An orange burst of light is a neural network that we form from one of these little spheres. So one of these little spheres, once again, is 0.5 millimeter in size. The orange color there is the calcium release during the synaptic process, and we're picking that up in a traditional flipper machine, okay? We'll let that run for a second. Now, you can imagine that's just one. Well, what if we look at all 384 of these brainwaves, we call it, or brainwave signals? Now you can see that here on this image as it builds. Now there's high functionality at scale and consistency. Now we had to converge that with augmented intelligence in our case because we were extracting out all this information. What used to take months for our scientists to analyze thousands of movies is now down to minutes. And we're able to do that through the integration of these data science techniques. Now, a little bit more about the characterization of Microbrain 3D, which is one of our platforms. You can see here the sphere. You can also see here the expression profile of key neuronal receptors. We've also done comparisons with human biopsy brain as well. We can cover that in the panel at the end if you'd like, but also showing that it's highly functionally active through different methods, whether it's multi-electrode array or through calcium oscillations on a flipper machine, for example. And the system is responsive to classic neurotransmitters that you can see here. Of course, there are many more that we have, but as an example, responsive to pharmacology. And then it's also reproducible in functionality as well. We've run experiments that have been months apart, and you get basically the same repeatable, reproducible information. So this is, you can see on this slide, and then of course the bottom right, great consistency in size. And once again, this is iPSC technology, so it is highly scalable. Here you can see a primary screen that we did with Johnson & Johnson or Janssen research out of San Diego, where we screened the LOPAC library. There was low coefficient of variability. And when you look at this paper, which you can actually see on +1, this is in Press, and this is a paper that we did recently with them. So hopefully you can search out this paper, and in the end, the conclusions were you can really use these models for discovery. And today we're going to show you in a little bit more depth on how we've been able to do it. Now, how do you create disease models if you're going to do discovery on these iPSC models? Well, there's essentially 3 basic buckets. The first one is patient-derived, so getting a sample of skin or blood from a patient, and the genetic information, the mutation that, let's say, you're studying, which we'll get into an example, flows through the process. So the plate of brain's organoids that you have on one of these plates that I'm holding up again could represent, has the genetic information and represents the disease. And we're going to go into more depth on what does that disease look like from a functionality standpoint. And then number 2, genetically engineered. So we're now in the age of being able to almost program biology or engineer biology with technologies like CRISPR. And then the third is inducement, whether it's through the environment, virus, tumor cells, perturbagens, you name it, those can be used to induce disease. Now, what's exciting is this is also a way where you can do some very interesting discovery and development. You can do activated target pathway and phenotypic discovery and development. So you can induce a particular function and activity in the model through the use of, let's say, a chemical to actually test your hypothesis on that model, and then do it in high throughput so you can test any combination concentration of that particular question that's being asked. Now, taking a step back a little bit, when we look at these, when we look at this capability of these platforms, each platform can be multiple diseases, a la the previous slide. Now, each disease can represent, can be many diseases and represent multiple drug discovery opportunities. And then each of the diseases, whether they are rare or common, can be made into these plates at high scale and high consistency. And then we can then test any combination or concentration of the disease in question. Now, how does this look like in terms of flow? This is, I think, an important slide. Many of you have these equipment on site, but here again is a high throughput, high density waveform of 384 waveforms. Now we're going to start digging in now. Here's a healthy waveform. If you zoom in on the waveform, then you can look at particular diseases. So today we're going to look at Rett syndrome. But in this case, I'm showing you the waveform here early. And then you can use the compounds that you want to test, drop them into each of the wells, any combination or concentration, into the wells to treat them and see how the response of the biology takes place. Now, you don't really want to be sitting in front of the computer screen for months at a time analyzing thousands of videos, so we do a lot of parameter extraction, things like frequency and amplitude. We can extract out from all this information very quickly in minutes. And then we can visualize that data like this in radar plots and like this, and you can see, okay, this is what the healthy looks like, this is what the disease looks like, and then this is how the parameters are adjusted through the introduction of new chemistry. And then you can plot it out. You can look at dose-dependent rescue and recovery across control, your disease, and treated. And then you can see not only is there good recovery, but also if there are side effects. And that's in a human model platform. And then what's also very exciting is then you can rank order. You can rank drug efficacy by target. And here you can see we've got a few targets here, multiple drugs, and we can show the recovery across various forms of parameters that we extract out. And early on in the day when we started, we just had a few parameters. Now we've got dozens of parameters, and we can look at them in every which way we can to bring forth the most useful information to make the best decision possible on what compound and clinical candidate should move forward. Now, we talked a little bit about mechanobiology before, and I know you've had some speakers in the past talk about the challenges in this space, but we can share that over the last few years, whether it's biowires or technologies such as this. Now, what you're looking at is in the bottom of a 384 well plate, we have created microtextured film that aligns cardiomyocytes into a sheet of fibers. And so on the right-hand side in the middle, you can see the previous two-dimensional approach, and you can see the formation of the green plakoglobin. It's just globbing up, and the actual cardiomyocytes in kind of a fried egg shape, not really looking like what's in all of us. So then comes where mechanobiology can be very helpful, and we have a product called 2.5D, we call it, where we can actually structure the cells in ways that we could more accurately recreates the biology. And you can see here also in green the plakoglobin on the distal ends of the cells, which is really critical for the electromechanical process of cardiac contraction. And so that's on the right-hand side, and you can see how far the industry's come. And this is just one of those examples on how we can recapitulate biology in a more accurate way. And then everything that I've said about microbrain or brain neurospheres is essentially the same for our microhearts. And here you can see the 384 well plate, 300 of these heart spheroids or microheart. The signals from those hearts you can measure also in parallel, like the brain platform system. And then one of my favorite videos out there is on a plate, you have a human 0.5 millimeter size heart that you can use for any number of uses. And so now we're going to dig in even more and look at an example of how this can be used. And in this case, this is on a rare disease called Rett syndrome, and it's a mutation of the MECP2 gene. And you can see the characteristics of this disease. And it's basically a neurodevelopmental disease. And we'll get into the example now. So when you look at this system side by side, you can have the microbrain on the left, and the RET version on the right. And they're similar in cellular composition, as you can see. Now, the model is also very reproducible. You can have the control and then the RET spheroids on a plate. You can see here what's being shown is a mosaic, if you will, of the disease. So RET, wild type, and mixture of the two. And you can see the impact of the sizes over a course of weeks. And this is where it gets quite exciting. So this is about, again, digitizing biology. But before I get into the digitization part of the biology, I just want to highlight that on the top, you've got the microbrain 3D control, and then the Rett syndrome on the bottom. And you can see by eye that it is very different in signal. But I can share with you that the many diseases that we've now done, sometimes by eye, you can't see those differences. And it really takes software technology to be able to eventually see it. So what we're doing here again is extracting out phenotypic information, extracting that out from the information, and then plotting it in ways. Now, once again, screen 1 and screen 2, it doesn't say here, but these were months apart, and you can see how similar they are in this in vitro human system. And as I mentioned, we've got dozens of biologically relevant features that we can extract out now. And then this is really the tip of the iceberg. So this target agnostic phenotypic screening approach, by looking at functional rescue, is the beginning. You can look at morphology, you can look at cell viability, and these are just peeling back the onion on answering question after question in a very effective way that brings a lot of value forward. Now, once again, you can see the concentration over different dosage. You can look at the features in the middle there, and then you can plot recovery across that concentration at the very bottom. And so you can take it a step forward. You can look at across the therapeutics. You can look at the cellular pathways that are being resonated in the system by the introduction of these of the new chemistries that are put on the system. And then on the right hand side, you can see control, what the disease model looks like, and then by color here, what recovery looks like, to middle of the road recovery, to even side effects. And then to dig just a little bit deeper, you can see in this case, looking at one of them, the cholinergic system here, agonists and antagonists, and how some of them show side effects and recovery, and then really understanding and seeing and realizing that it's really the acetylcholine esterase inhibitors that are predominantly showing positive good recovery. So you can start digging deeper and investigating these questions, which is extremely interesting and exciting. Next, we're going to talk about clinical translation, which is an interesting, I think it's going to more aptly answer the question that was asked, Tom, by the listener today. And we're going to go into a couple examples. One is around neurotoxicity. This is some work that was done with Takeda. They're part of the IQ consortium. They screened 84 compounds across multiple mechanisms of toxicity, looking at compounds with frequent to low reported events, adverse reported events, and then you can see how they use this model with classical regression approaches to understand what the specificity and sensitivity was of the system across being tested in a blinded fashion across nearly 100 compounds. And we're very excited to show that the specificity scores were extremely high, as well as the sensitivity scores. So this is hopefully answering part of the questions that now there are certain tests, let's say from a looking at early hazard identification, looking at neurosafety compounds, perhaps this is a better model. And what we're hearing is in the industry and seeing is that this is now being used and replacing some aspects of what would traditionally be used. So we're very excited about that. Now, what about predicting efficacy? Now, here is a compound, Anavex 2-73, that's in phase 3 clinical trial right now, and it's a sigma-1 receptor agonist. Now, we screened a similar kind of sigma-1 receptor agonist in our system, and we saw efficacy across the dosages. And you can see, once again, by eye, you can see the control, the Rett disease model, and then the examples down below across the rows of how by eye you can see the recovery in the waveforms, but then also see it on the right-hand side with the parameter recovery as well. And so this is a very exciting way of doing this and doing it in a human model system and getting and screening hundreds of compounds potentially, or whatever the interest is to answering the question that you want. Now I'm going to get into the now and into the future. I'm checking the time here when I look down, but I think we're doing okay. So there's a few examples I want to jump into about now and into the future. And it's going to start off with what we did back a few years ago on Zika virus. So there were potential repurposed compounds that could be used to combat Zika virus. And one of the questions we had was, well, do these potential repurposed drugs have an adverse effect on unborn or on pregnant women and their unborn children? And so when you look at that, you can see here at the top are actually progenitor cells. So these are not IPCs, they're way further along. They're almost neurons, but these are a stage before mature neurons. And you can see in the color here in viability that the orange is low viability, and then blue is high viability based on percentage. And you can see on the top there that there's a lot more orange on the or low viability of the progenitor iPSCs versus mature. And so this really brings forward a question of, well, can we use these technologies in ways we could never do or would not do in clinical trials because of vulnerable populations, pregnant women, children. We wouldn't do that. But here, to decrease the risk, especially for vaccines, because you want to do no harm with vaccines because you're giving essentially that to healthy people, how would you determine safety? Well, this is a way to determine safety in a novel way without risking anybody's lives. So here you can see new capability to test neurodevelopment, and then of course showing the differences in how cells behave based on maturity. And then looking again with the background of information that we have now, how we can use the visualization of data to see what control, safe, and then unsafe response looks like in a very simple plot. It's much more complex now, but this is to give you an example of how this can be done. And then we also did some work with US AMRID on infectious diseases or weaponizable viruses, testing everything from Ebola, dengue, Zika to EEE, which is now going across this country, unfortunately. And this is an example of how not only can you look at infectability of the model, but also use the model as an inhibitory model, a human inhibitory model, to find vaccines. And so that's a very powerful usage again. Instead of using, let's say, HeLa cells or animals, this is another way you can use this platform to, in another use case. Here's another use case, and this is something that's simple. It's not introducing a perturbogen, but it's using or gene editing or anything like that, but it's actually creating a concussive event on the spheres itself. So this is some work by Dr. Finan at North Shore we've done with him and his team, showing how you can concuss these plates to create like a CTE event and then study that. And then, as you can imagine, the variables explode if you want to see how these capabilities can intersect various diseases. How does concussion affect a cell line with a particular disease, for example? Here's another one. Glioblastoma, high throughput orthotopic tumor model on a plate. You can see day 1, day 3, day 5. This is a highly mutated line, but I'm going to show you a really cool example on the next slide. But just to as a conceptual slide, I want to show you this one first. So we fluoresce using mCherry the tumor cells that you can see in the reddish color, and they have attached onto the neurosphere. And it took some invention to figure out, some innovation to figure out how we attach and keep the tumor cells alive long enough to have them infiltrate the neurosphere. But you can see it happens very quickly. And over time, they begin to integrate into the sphere system. And I'll show you on the next slide. But just to lay the groundwork of this approach, we're looking at here 4 rows, right? You can have a system where it's ineffective and safe, right? So the neurosphere is fine. You can see the graph of the neurosignal that we were showing earlier, and then how the red cells are growing, so it's ineffective. And then there's also, but the sphere is okay. But then there's also situations where it's ineffective and toxic. And then there are situations where you have effective and toxic, and that's not going to work for the patient either. And then lastly, you could ideally have effective and safe. And so just laying the groundwork here, looking at a cancer model. And then here, once again, you can look at this human orthotopic tumor on a plate in 384. And then you can look at the different stainings we did to show where the tumor is on the sphere itself. And then also we've done cryosection of the spheres to see where the tumor cells are inside of the 3-dimensional space. We then recently did this work with BMS Celgene, and here's a poster that we recently showed in a public setting, so I can show it here. And this was showing how they took patient tumor lines and introduced them into our system, and then showed how you can quantify and discriminate and assess the tumor line infiltration, proliferation inside of that model, and then to look at the potential therapeutic impact on that model, and then also potential adverse effects. So if you have interest, reach out, and this is something we can show or go to the website. But once again, this is an orthotopic model for drug discovery, but what's near and dear to my heart, especially because I have friends who've had family members with this disease, is this opens up the way, a future of how not just looking at tumor cells that have been biopsied from a patient, but putting them into a metastatic model to understand how we can stop the progression of a particular disease, in this case, glioblastoma, in a neurosphere. So if you can take that step further into the future, you can imagine one day when we all go to the hospital and see, let's say, a general practitioner early in our lives, and they get a sample of skin or blood, and it's like, oh, okay, like we're gonna go take your biopsy and then make you on a plate, right? You're a person on a plate kind of example. And when the time comes, test compounds that are out there, or even experimental ones, on what might actually work for you. So this really opens up many doors in how, on a personalized level, these technologies can transform how we approach human health. And then to bring in the example from previous slides that I covered today, the usage of AI drug discovery in combination with everything that we do, everybody on this call, and I just want to lay this out. So you got the low-hanging fruit of potentially these integration of this metastatic model. You've got the now very popular AI drug discovery. Now the challenge with AI drug discovery is, yes, you can have tens of thousands of recommended potential chemical structures, but how do you test those 30,000 chemical structures? And having an in vitro system to rank critical to advancing this field forward in this new paradigm shifting way.
Ping, I'm sorry, can you take a step back for about 20 seconds? Your audio and screen froze.
Okay,
Sorry about
so
that.
no problem. So do I need to go to start this slide again, or?
Yeah, I would. I
Okay,
think that'd be helpful.
so what's really powerful here is that the novel is using novel biological models such as this GBM model, glioblastoma model, but also integrating it with things that we've talked about already today to give you the whole picture on how it comes together. AI drug discovery, which is very popular, but when you screen that many compounds, when you have, let's say, tens of thousands of recommendations, after you in silico screen billions of compounds, you're still left with a situation where you have 30,000 compounds and you don't know which one is the one that you should bring forward. And so it is critical for in vitro models that we've discussed today to be able to screen thousands of compounds and then rank order them to see what should move forward, but also the in vivo aspect, the seasoned, tested, battle tested over the last several decades work that many of you have done also becomes critical. And it's this combination of novel biology, cutting edge AI drug discovery and silico approaches, data science, and all the things that I've covered today in combination with these in vitro models at scale that then integrated with in vivo approaches is going to accelerate drug discovery and have kind of a best of both worlds, of all worlds, if you will, where the Venn diagram where we want to be is covered by all groups. No one group maybe covers for each of us, but when we all come together, you can have accelerated drug discovery, but also de-risking and also savings over conventional costs. And that compression begins to be realized. And then I think lastly, this might be the last example before we get into tie things up here. Our scientists, our most recent work by our scientists have been on the cover of Cell Stem Cell late last year, and they made a complex cortical organoid model that you can see on the left. And essentially, if I were to describe this to my 10-year-old daughter, I would say, if she said, well, how is this different from the previous model? And I can say, well, our previous model was like a chocolate M&M. If you cut it in half, it's a solid, piece of neurons and astrocytes, all chocolate. But here, it's like a peanut M&M. Inside, you can have a subcortex. You can have, as our scientists explained, layered stratification inside and unique structure formation within the neurosphere body. And that's what our scientists are world experts on. And Discover magazine actually identified as one of the top 10 discoveries of 2019. And one of the reasons why is because of the graph on the right. By looking at the local electrical signals of the organoid itself, it actually compared quite similarly to the EEG of a newborn. And so they're looking at these, what are called SATs, spontaneous activity transients. And you can see by eye the uniqueness here. And so now we're bringing together an example here of how the world of human organoids begins to overlap with human biological function in some very meaningful ways. And the industry is progressing very quickly in doing so. So in summary, what we've done at the company and with this team is converge the world of human organoid platforms with software analytics and AI machine learning, with an ability to do drug discovery in high throughput. And in concert, allows us to do things at speed, with accuracy, and to look at new targets in a human system, to really unlock some of the most challenging diseases that are facing humanity. And through the work here, and maybe with all of you, make cures possible. And so in summary, we've put together this slide here on the left, just showing the highlights of the talk today and how these technologies are transforming drug discovery, how preclinical timelines can be compressed, how human insights can be achieved pre-animal, the combination of biological models and in silico can lead to better predictiveness, and how these technologies can open up a vast new areas of therapeutic discovery, and how you can actually de-risk medicines for vulnerable populations. And what's exciting with all of you is having this conversation of how all of these things can work in synergy with the 3R principles. And once again, I'm showing the graph I showed earlier, just to reiterate and summarize these, how we bring it all together, and how the combination of all these new technologies, together with the work that all of you are doing, can compress timelines and bring medicines to patients faster, safer and more effective medicines faster. So with that, I will pause, and we'll open up the panel, which bring the real PhDs onto the panel, I guess, and I'm excited for the Q&A session. So thank you very much. I think we're at 12:59 or 1:59 East Coast time. So let's begin the panel.
Thanks, Ping. I think I've sent all of you some questions as they've been coming in here. I don't know if you can see them on your screens there. I'm happy to read them, or if you guys want to address them yourselves, go right ahead.
I love the questions. There are many of them, and hopefully some of them have been already answered by the folks. But if you want to MC this, we can cover it. How do you want to approach this, Tom?
Sure. I'll go to some of the beginning ones here and work my way down.
Well, I'd like to say hello to Blake, Barb, and Glenn. We are very excited about this merger that was announced, and I think you'll find that this group brings together experts. Blake, who spent his career in induced pluripotent stem cell technologies, is one of the first to lead teams to do so. And then, as you saw, the bios of Barb and Glenn, we're super excited to have them join Blake and I, because we really believe that the synergy of these innovative technologies with these in battle-tested in vivo approaches are really going to help change the industry in a way from the inside. So we're very excited to have that conversation with you today.
Great. Thanks for joining us here on the panel. I'll just go down some of the lists here of questions that have come in. You talked about the potential for animal replacement. Do you have any examples that point to numbers or percentages related to such reductions? Or I think that's speaking to the any real world examples from any of the testing.
I think I covered a little bit of that. I mean, it's beginning. I think it's beginning to tip. I can share an example just with the conversation with the big pharma executive, and I said, what would you do if you didn't have these plates? And it would be, well, we'd be doing a lot of animal testing on that, and we don't have to do that now. But I think it's you can see this is just the beginning. Whether it's neurosafety, you can imagine going to different areas, and maybe I'll open it up to the panel since they're here on where they see the replacement initially beginning, but where it could go.
Yeah, Ping, one thing that we can also bring up along the lines that you'd mentioned is, the example that was given to us of I believe it was a retrospective study where 20 out of 22 compounds would have been caught for toxicity in the microbrain. But instead, because that preparation wasn't available at that time, those were not caught until after animal studies. And so you might think one of the first areas that this is going to be, find that prevention is simply in negating the need to get those poorly performing molecules into animals. So.
Yeah, and then into discovery. So you could recreate through the various ways of creating disease models that we covered today, and creating those disease models in vitro, and really investigating ways that you wouldn't necessarily do through primary cells. So traditionally, primary cells aren't going to come from individuals under the age 20, for example. And so when you're talking about neurodevelopmental diseases, you're just not going to have those samples to do discovery work. And in these models, it opens up multiple options and the hundreds of rare diseases out there that this now enables.
Thank you. Next question I had on list here, as the organs get closer to being real human organs, the better their predictability will be. And can you speak to the advancements that are being made with the induced pluripotent stem cell technology to get these models closer to more complete organ systems?
Well, I'll take a first swing, and then I'll let Blake add more color. I think where it seems like every 6 months there's new technology coming, and I think not only are there organoids, but it's the intersection of organoids with other technologies to better recapitulate biology, whether it's wires or multiple fibers on a surface, or assembloids, or and maybe we'll let Barb talk about tissue on chip technologies, because she's an expert in that space. I think that yeah, I think the sky is the limit as the intersection of engineering and biology begin. So I'll pass it to Blake and Barb to add some more color.
Go ahead,
Barb?
Blake.
Okay, all right. Yeah, I think you alluded to that in one of the last slides, in that the complex organoids coming out of Alison Motri's lab. We're going to be seeing that both the cellular complexity and the structural complexity of organoids increasing and bringing in more and more biology. And really where we come into play there is that with our scientific background combined with the innovative aspects of the academic field, really taking that art and transforming it into an industrialized setting. So just like what we did with the spheroids, we took it from really an interesting phenomenon and expanded that into a high throughput 384 well repeatable platform. We will be able to continue to partner and take those next steps in advancement and move them in. And that's what we're doing with a lot of our pharma partners right now.
Yeah, and I'll just add to that. I think that one of the greatest promises of the organ on a chip is that you can create different environments, and you can do some downstream testing and of metabolites, and analyzing for metabolites and things like that. But it's important to say that that technology, I don't think, is in a place where you can use it in a high throughput manner. And I don't think that we as an organization or as a group would be comfortable taking it there until we're ready to do it with the same rigor and at the same standard that we're able to do the organoids. So you may see you may see it develop in that direction, but I think that that's probably a bigger limitation to the organ on a chip than what we have with the spheroids in the well format right now.
Yeah, thanks. Tom, I think I see the next, there's quite a few questions, and I want to be cognizant of everybody's time. So the next one I think is around, is the FDA validating these technologies in place of animals in some of the early stages of safety testing? Is that the next one we're on?
It is, and that's, I think, a good question for this group. We've seen this similar questions previously, so I'd love to hear this.
Absolutely.
I can answer this, Ping.
Yeah,
if you
please.
Want me to.
Yes. Yeah,
Okay.
Barb, please.
All right.
All right.
Blake. Sure, sure. So the FDA has a division of applied regulatory science, and they're actually working in this area quite diligently right now. They're working in it with an international consortium on pro-arrhythmia assay in cardiomyocytes. So they're looking at validating that technology. They also have an initiative in integrated cellular systems where they're looking at hepatocytes and cardiomyocytes and trying to come to that. The question, so there is work being done, and of course NIH is in their translational science area is looking at clinical trial on a chip. People are really looking at this hard, but I think what we all need to think about and expect is that this is an additive technology. It's not likely to replace animal studies in the near term. And that's very understandable. I mean, even if we got to a point where we were absolutely sure that we were detecting a specific condition or adverse event, how comfortable is everyone going to be with saying, okay, but we don't need to do any animal testing? We all hope that it gets there someday, and we're doing our best to make it get to that point. But I think you should look at this as an additive, as Blake said earlier, a way to make sure that you don't take anything into animal studies that aren't going to be de-risked through the human first approach.
Blake,
Did you
anything
have
to add?
anything else?
No.
Yeah.
Okay. Barb covered it broadly and succinctly.
Good.
Barb, shut up.
Lots of questions here. So, how far away is the technology for use in personalized approach? I think it's happening now. I think we're at, once again, a renaissance of not just what we talked about today, but the genomic revolution, the whole genome sequencing combined with these technologies, the big data information from populations integrating with these in vitro approaches to run actual experiments, as opposed to sometimes big data, it's big, but it may have quite a few holes in it. And so this allows you to have an in vitro experiment to answer key questions. And then of course, yes, I think tumor cells are already, being formed into tumoroids to answer questions on what would be best for the individual. But our point is that what if you were to integrate that into more of a system that is more biologically similar to what's going on? And I think so. It's happening now, actually. I don't think it's far away at all. Next one. Can you expand on the value of basic research in forms of drug development? And this one might be right up the for Glenn here, because he's had many years looking at these questions from different angles. And so from a basic research perspective, Glenn, how do you see this informing drug development?
Yeah, I think they can contribute during the whole continuum from target identification and validation all the way through to safety assessment. Certainly with these organoids, we can manipulate, we can knock out certain targets that might be of interest for target identification. There's ways of validating those then by looking at, okay, if we can chemically manipulate that pathway. So very early on in the discovery process, then once you have those systems in place, then they could actually apply in the lead optimization phase of a typical drug discovery program. And again, that might be one of your R's, where if you can replace standard rodent pharmacology models for lead op, I think that would be a great contribution in that stage too. So I see them, again, relevant from beginning to end.
Yeah, and I think there's a part two here, which is maybe giving an example of how these new technologies and traditional animal work could be integrated in to create the most predictive value by bringing these together. Maybe you can shed some light on that.
Sure. Yeah, I think with the microorgans, Again, we're working in a human system, which is certainly powerful, but there are limitations that will not replace whole animal pharmacology studies. Kind of your proof of concept efficacy work that you need to do at the end of your lead optimization. If you're picking your development lead out of your final set of candidates, you're still going to need that in vivo pharmacology model, because you also need to set that dose range. So that when you're looking at your toxicology studies, you have your toxicology, your safe dose range, so you can set your index, your safety margin. Those things aren't going to be replaced by any organoid in a plate, but I think they're completely complementary. And again, the strength of using these as kind of classic pharmacological approaches of inducing disease chemically or whatever, looking at antagonists by providing an agonist to the system, vice versa, looking at agonists. I see them really complementary to the in vivo. Specific example, there's so many. I think there's so many ways if you're looking at, especially a classic small molecule like a G protein coupled receptor target or an intracellular enzyme target, where you can really, I think it's straightforward to see where these models can really help you reduce the number of animals you need to run through in optimization, and then in final candidate selection, and when you're rank ordering your dozen, your 10 candidate compounds.
Great, thanks, Glenn. And then lastly, before we open it up, the last question here on this list that Tom you forward is, can we speak to how cell maturation is addressed within the model to ensure better predictability? I think that in all our models, but taking it from a conceptual standpoint, what was enabled was that we chose a cellular system that was diverse enough to essentially be matured. And that maturity over the course of many weeks could be achieved because of the cell types that we had in the system, meaning that they could metabolize the nutrients in the media and allow it to function biologically for long periods of time. And so we often look at things like chronic testing and aging of these lines to look at various diseases. I think that one example is that when you bring, let's say, X number of neuronal cells together, they just don't instantly form a neural network. It does take time, and I think that approach, because it was enabled by the cell types that we have, to actually mature these models, then created the neural networks that you saw. It wasn't just individual cells or individual cells that weren't communicating with each other. It was actually a neural network that was formed, or in the cardiac example, cardiac contraction, and looking at that system in similar ways. And then as you age them, you can see these more mature function. And from that more mature function, you then have a signal that you can observe and then probe through chemistry to see how that signal changes. Now you can, and that's one example of that, but not just function. And as I shared in the presentation today, that's kind of the first target agnostic approach of looking at biology, but that's just really the first layer of the onion, if you will. You can keep peeling it back and look at gene expression and all different aspects that our scientists are really good in, and to answer questions after you get insights on overall function. So those are some ways on how maturing the function is really maturing the underlying biology, which again is showing matured structure as well. And all of this plays in concert to getting more predictive answers to the questions that are being asked.
Thank you. I do have some comments that have come in when many thanking all of you for your time and for your presentation today, and comment about your enthusiasm, which is really great to see. Another comment about the interplay between the technology and existing animal work for better prediction comment, near and dear to our hearts here at NJABR with our Homes for Animal Heroes Retirement Act. The comment that this is important because fewer animals will be terminal and more will be able to be rehomed following testing. So that's always something we're looking out for
I like
here.
that term, rehomed. I love that.
Rehomed, yes.
I'm
A fourth R.
Yeah, that's the fourth R. So we said rehomed.
Love that.
That's really cool. That's really cool.
Writing it down.
Yeah.
And,
Well,
and
yeah.
I also want to address for some folks who did get here late. Yes, the webinar is being recorded. We'll get it cleaned up and out to all the registrants as soon as we can when we are done. And hopefully before the week is out, we'll get that out to all of you.
Well, thank you, Tom. We really enjoyed the conversation today. And I think the excitement comes from seeing the potential of these technologies and what difference it can make in millions of people's lives around the world. And, you know, their brothers, sisters, fathers, mothers, you know, children, I mean, everyone is impacted. And we really appreciate the opportunity to share what we've been up to this community, and hopefully we can work together one day. And if so, feel free to reach out. We'd love to do so. And thank you so much, Tom, for the time, and to New Jersey Association for Bioanalytical Research in making this possible today.
Thank you. We appreciate your time, Ping, and as well to you, Glenn, Barb, and Blake. We thank you for sharing your expertise with us. Just real quick, I did want to advise again, if anybody has questions they'd like to follow up with Ping directly, please go ahead and do so. This email address is right there on your screen. Then one final comment for everyone. We have a few weeks till our next webinar, which will be on November 11th. Featuring Dr. Reggie Gazes with Bucknell University, who will be speaking on a presentation entitled Automated Cognitive Testing in Socially Housed Monkeys, Implications for Research, Care, and Management. Again, everyone who is new here, this is part of a larger 3 Rs webinar series. Each webinar has been approved for 1.25 CE credits for a total of 15 for those of you who are able to participate in all of them. For more information about the series, please visit the URL that's on your screen right there. And I'd also like to make sure I thank our colleagues at the North American 3 Rs Collaborative for their support and help in executing this webinar series. So again, we'll see everybody next time. And Ping, Blake, Barb, and Glenn, thanks again for all of your help. This was really great and enjoyable,
right,
and we look forward to continuing to partner with you.
Sounds great. Thank you.
Nice
Take
meeting
care.
everyone. Take care.
Bye.
Bye-bye.